Hello friends, welcome to our next class of Principles of Inheritance and Variation. Now today, my dear students, we are going to understand the terminology multiple alleles. What is the meaning of this term? The characteristics with examples. We have heard this word allele, and if you remember my last video, you have seen and you have understood, I believe, that every gene which is placed in a certain locus has two partners. And we know that every gene exists in homologous chromosome as homologous genes and in pairs. For the same character, one gene is from the paternal side and the other gene is from the maternal side. And we say that for character X, for example, there are two alleles. Now, most of the characters, the phenotypic characters or any of the characters that are expressed, are controlled by such pair of genes known as homologs alleles. But then in nature, something different has also happened. Over thousands of years of evolution and change through mutation, we find that today we come across certain genes which exist not only in two alternate forms but more than two forms, three or more. Now, such genes which have more than three or more alleles are known as the multiple allelic genes. Now the question is, we have learned that for a gene there can be only two alleles in a particular person or an organism. What about the third, fourth or the fifth allele? It is something like this, that in my color pencil box I have got 10 colors, but I can at any moment of time choose only two. So I can make 5 pairs of different combinations, and all these pairs would be expressing the same character, however with variations. Now the character could be skin color, there would be variations in degree of fairness or darkness, or it could be something else. We shall come to that and we'll learn in details with examples. Now as you can see here, that the word allele, it denotes to alternative forms, as I said, to the two forms of genic pair. But in case of multiple allelism, three or more of such genes occupying the same locus. And what does that mean? It is very important to note that unlike separate genes, all the alleles of a certain gene would be always in the same position of the same chromosome in an individual. Say in chromosome number 7, in locus number 2, there is a pair of gene for character Y, but this gene is present in 5 forms. So in one person, 2 of them will there, in the other person another 2 of them can be there, but never ever they will move away from the chromosome number 7 or locus point 2. This is important to note, that every multiple allelic gene has to be in the same locus of the same chromosome in the homologous pair. Anyways, this has produced a wonderful variability, and sometimes if we are not aware of the commonness of the gene, then we might confuse it with being a variation at the species level. Now I shall like to give you some idea about what are the important characteristics of the multiple alleles. Now, as I've just said, multiple alleles are situated on homologous chromosomes. They have to be. They have to be in the same pair of chromosomes which are homologous. And you know by now what are homologous chromosomes. Those chromosomes which are of the same size, having their centromere at the same place, but have been acquired one from the father and the other from the mother. So the genes of multiple allelic types will always be located in the homologous chromosome and of course in a particular locus position. And since it is so, there is no crossing over, because when there is gamete formation, the crossing over that takes place between characters is always between intergenes. There is no crossing over in between the intragenes. I may have a pair of pencil, a pair of color pencil, one red and one blue, and I can have a pair of chalks, again one red and one blue. I will not have a breeding between the pencils or between the chalks, but I will have a breeding of the pencils with the chalks. My red pencil and blue pencil will not become half red, half blue, but it can become one pencil and one chalk. That means we have to go for intergenic combination in case of crossing over, and therefore, since multiple alleles are located in the homologous chromosomes, there can never be crossing over amongst or between them. That is a very important fundamental quality and character of multiple allelic genes. And of course, as I told you already, that multiple alleles influence the same character, albeit with variations. Now, these variations are subtle, sometimes pronounced, but you have to be more focused and conscious to understand whether it is some other gene or it is the same gene which is playing a phenotypic trick on us. Now, with that, it is also a characteristic that we can use the multiple allelic genes and use our statistical biology and find out the population pattern and how it fans out in the different population. Well, multiple alleles will never show complementation with each other because they're in the same house, they're in the same place, and they are basically the same character. Complementation between genes is basically intergenic interaction where one gene of character X influences another gene, maybe in another chromosome of character Y, to produce a product which is Z. That is complementation. We can always like a food with proper salt because the salt complements the fry. Maybe you are trying to have a good fried potato chips, you require a pertinent amount of salt to make it more tasty. That is complementation, but that has to be between the salt and the potato, not the salt and the salt itself. That's what is called multiple alleles can never show complementation to each other. And remember, a very important feature of multiple allele is that it must have a primitive wild type. The terminology wild type denotes the first form of the gene which was present, but it had undergone evolutionary changes or mutational changes and has formed different types. So there has to be a fundamental gene and then subsidiary genes. Those subsidiary genes are the alleles of those genes. Now, normally, a wild type has one allele. We call them allelic pair. But sometimes one wild type has 1, 2, 3, 4 alleles, and that's what makes it a multiple allelic gene. Again, we find that in F2 generations, the multiple alleles do follow the monohybrid ratio, thus telling us that they are but in the same homologous chromosome. Now, I believe that you have understood the basic characteristics of the multiple alleles. Now, there are various genes in nature which are multiple allelic. I've just listed 4 of the examples. You can see one is blood group, which is the Kerr-Landsteiner's ABO blood group. Coat color in rabbit. By coat color, we mean the color of its fur. Also, the coat color in mice is similar to the coat color in rabbit. And the eye color in Drosophila melanogaster, which is the fruit fly. If you have a banana at your home, or if you have a piece of lemon, you will find small flies, insects come and sit on those. So those are the Drosophila, and they have different eye color. They could have red eye color, black eye color, amber eye color, and all of these changes are basically because of the multiple allelic expression of the same gene for eye color. Okay, so I'll explain to you multiple allelism with 2 of the most important examples. One is the blood group in human being, and the other is the coat color in rabbit. Because if you understand even one properly, everything else follows the same principle. So for the blood group in human, we are aware, and these days people are most aware, that blood is said to be having a certain group. Almost everybody of us are aware of my group is A, my group is B, O, AB, or also we are in the habit of saying I am A positive, I am B negative, I am O positive. So what does this really mean? Now let me tell you that there is a gene like every character. The character blood group is also controlled by a gene, and this gene is denoted by the letter capital I, which stands for the isoagglutinogen. Of course, it is another name of antigen. Now, what is it? It's a protein. So this protein is expressed by a gene, we call it I, and we have already understood that a portion of the DNA which can express a protein is a gene. Therefore, for our convenience, the gene does not know we call it I, but for our convenience, we have denoted that certain area of the gene of the DNA as gene I. Now, the gene I is responsible for the protein on the surface of the red blood corpuscles, the RBCs. The protein type, what type of protein it is, will really determine what type of blood group we have. It is very simple. Now, this gene I is a bit tricky, and what it did in the human population, it changed its nature into 3 types. It became IA type. You can see we have the capital I with a superscript A IB type, and a small I or a capital I with a superscript O. You can use any of those interchangeably. Small I because it is recessive in nature, but the capital I with a superscript O is actually null, means no antigen is there. It's 0, null. And accordingly, we have denoted our blood group as blood group A, blood group B, and blood group O. That means, well, what does it mean? Let us see. Now, if we analyze the thing, we'll see that the mutant allele IA is equal in its potency and co-dominant with the mutant allele IB. You can see the relation I have given in bracket in the fourth line, where A is equal in its potency to B, but both are greater in their potency to O. That means IA, which is a allele for blood group or blood protein, is codominant with another blood protein IB, but individually both of them are powerful over bechara small I or capital I with a superscript O. Now, keeping that relation in mind, if we try to understand, then what genotype will create what phenotype? Or rather, if I hear that someone is group A positive, then what genotype that person can have? Let us examine. If, as in serial number 1, if a person is having a homozygous capital IA and a capital IA, that is one pair of allelic gene, because there will be always one pair in any homologous chromosome, or the person is I capital A in one and the other allele is small I, the person will be type A, because in the former case where both are A capital I capital A capital I capital A, that means both are IA, so both are equal, both are same, they will create group A. But in the next case, when IA is combining with small I, or for that matter capital I O, bechara capital I is recessive, so it will again have A, the law of dominance, first law. In the second number, you can see the same is being repeated for group B, where homologous IBs and IBs will create a B, or a heterozygous IB and small I will create a B again. Only and only when from father side and mother side someone will receive IA and IB, then both will be equally dominant and they will share your blood cells 50-50. Some blood cells will have A antigen on their surface, some will have B antigen on their surface, and the person will be known as having blood group AB. But what about the last? The person who has no antigen, that means small i's or the I with superscript O, cannot produce any protein on the surface of the red cell, and therefore this person or such persons are said to be of type O. That means blood group O. Now, isn't it interesting that only one gene, wild type I, by changing its phenotype, by changing its genotype through mutation, has given rise to 3 different types of alleles, and in any given moment, in the body of any one human being, only 2 can exist. We cannot have 3 all them together. So we'll have a permutation among these 3, either 1 and 2 or 1 and 3 or 2 and 3. Likewise, we'll have changeability and permutation of those 3 in pairs of 2s, in pairs actually. So that is how our blood groups are different. But does that mean since our blood groups are different, we are different species? No. The gene is the same, commanding the same character. The only difference is it has got its own variation. Here you can see a wonderful table that I want to share with you, that if the parental phenotype, what group the parents are known, then the genotype can be found out, and if the genotype of the parents are known, then the phenotype and genotype of the children can be predicted. Say a person who is having blood group O gets married to another person with blood group O. Since they are O, we know that they have to have small i's in their genotype. They cannot have capital I. No way. So since they are O, they will have pure genotype homozygous small I for both the parents, and the children these two parents will create will be 100% O and will have a genotype of small I. So it is like a future prediction which is perfectly 100% scientific and accurate. We can take any of the cases. Say for example, we take the last case where a AB person is married to a AB person. In such a case, the father, if you consider one is the father and the other is the mother, then the father can either donate A or B, and the mother can also donate A or B. It can happen in any of those cases. So the children could be either A group, because it can have one A from mother and one A from father, so it will be A. The other children could be B from father and B from mother, so it could be B. Or it can be the third children could be one A from father and the other is B from mother, so AB. So parents who are AB will have children who can have either A group or B group or AB group. This way we can accurately predict and tell how the multiple alleles act and how they express their phenotype in the organisms that they are present in. Well, now I shall come to the next example. Again, a very brilliant example which is interesting too, and is the coat color in rabbit. Now I have told you that coat color denotes the color of its fur. Now, we might be very much aware of only the white rabbit. Most of us have seen the white rabbit, and in pictures and all, the white rabbit is most famous. But the wild type rabbit, or the hare if you may say, is called the agouti or the full color rabbit. Now here let me tell you that the full color rabbit, if I take one hair from the body of that rabbit, that particular hair, it is HAIR hair, because the hair from the hair, that means HAIR of the HARE. Isn't it interesting? So what we have, the strand of hair is one part yellow, one part black, and one part white. That creates a brownish color which is known as the agouti or the full color. The other form of rabbit that we see is known as the chinchilla. Yes, the name is chinchilla. And a single strand of hair from the coat will tell us that half of the strand is white and half of the strand is black. So it has a silvery color, like kacha paka. It has got half white and half black. So you have a silvery coat and is known as a chinchilla pattern. The third type of rabbit that we come across is known as the Himalayan, in which the rabbit is all white except for its tips. The tip of the tail, the tip of its ears, the tip of its paws and legs are black. Rest of the body is white. And then there is the last category of rabbit, which is pure white with reddish eye and pinkish nose. It is absolutely white. These are known as albino. That means they don't have any color and the strand is white. Now, for coat color in rabbit, we have a gene denoted by capital C. Now the capital C gene is a wild type gene. But this capital C gene over time had undergone mutation and has given rise to 4 different types of alleles. Earlier in blood group we have seen that the capital I had become 3 mutant types, but here we are finding that there are 4 in total of types of the gene C. And the relation is Here capital C is dominant over capital CCH. Now the CH stands for Chinchilla. And both of them are dominant over CH. H stands for Himalayan. And all of these three are dominant over the bichara small c, which is the gene for albino. Now we also know that for every organism there has to be a pair of genes for a particular character. So what we see in case number 1, where the rabbit is agouti, that means full color, what could be the genotypic combination, or what are the possible genotypic combination of the rabbit? It could be capital C, homozygous dominant, so it will be agouti. It could be capital C and capital CCH. Since capital CCH is recessive to capital C, again it will be full color. It could be capital C and capital CH, again the same follows, it will be agouti. Or it could be capital C and small c. In all of these 4 cases, the presence of capital C will cover all other characters and the expression will be agouti. But law of dominance, not pure agouti, except the first one, which is capital C. The other 3 are heterozygous agouti. In case of Chinchilla, we will now remove the capital C, because if capital C is there, you cannot have Chinchilla. For Chinchilla, you should have a homozygous CCH or a heterozygous CCH or CCH-C, because the rule of dominance follows true here also. In case of Himalayan pattern, again we remove the first 2. So CH becomes dominant over the small c. And what we can have? Either we can have CH or CH-C. That would give us a Himalayan pattern. And only when all of those capital C's are absent, and in both the locus of the same gene, the alleles are small c and small c, the rabbit will be white without any color, and we will call it the albino. That means colorless. Here, for your convenience, I have given you the pictorial depictions of those types, the wild type or the agouti, and their genotype. You can see in capital italics, the genotypes are given, either CC or CCCH or CCH or C small c. For Chinchilla likewise, you have the silvery color, and in the full color you can see it is brownish, yellowish, black, a combination of yellow, black and white. That's what makes agouti an agouti. But in Chinchilla, you have a black and white mix, and therefore the genotype is, as you can see again in capital italics, which is CCH, CCH and CCH small c. Coming to the other 2, you can see the Himalayan with its extremities black, the tip of the nose, the tip of the ear and the paws, the 4 legs and the hind legs, but the body is white, and the genotype could only be CH with CH or CH with small c. But the most beautiful is the white rabbit, but which is genotypically the weakest and is basically a double recessive small c genotype for coat color. And that is how all of these 4 types have been created without changing the basic gene for coat color, which is capital C, which is C. The C is the better word, because if you use for capital C, it seems that the capital C itself is a factor, but here all are factors, but the wild type is capital C, from which we had developed, or nature has developed all of these types. collectively, you can look into this chart where you have all the 4 types, the wild type with a brown fur, and what can it have? Capital C, or any of those 3 combinations from the upper bar with a capital C. The combination should be always thought of like this: the first bar and rest of the 3. For the second, CCH and the rest of the 2. For the Himalayan, CH and the last. And for albino, only the last with itself. So that is how a single gene called the C gene for coat color in rabbit had mutated, and mutated into 3 forms, totaling with the wild type 4, and that is what is known as multiple Allelism. Now, a layman could have looked at those rabbits and said, look, there are 4 different species of rabbit of different colors. But that would be very foolish genetically, because these are the same rabbit interbreeding and are of good species. But because of the trick genes play through multiple allelism, their coat colors have changed, as also the blood group in human beings have changed, as also the color of eyes in Drosophila have changed. And for that matter, there are many more examples in nature which makes us understand that yes, a particular gene can have its different forms, but commands the same character and remains in the same place, in the same locus of the same homologous chromosome. That is the beauty of multiple allelism. Now, to sum up, in nature, there are many such genes that started from a wild type. I've told you that already. That started from a wild type. But over the time, mutated or evolved into different traits. But remember, of the same gene. They have not deviated and become something else. They have not changed their fundamental nature of commanding the character they were commanding. However, they have changed the expressional degree. The command is blood group, the command is coat color, but variation in the degree of expression. That is the degree of expression with variation in expression. But not that a coat color gene has altogether changed into structural gene, or maybe your fundamental skeletal structure gene. That cannot happen. That would be a very deleterious or a very strong mutation. However, such genes of multiple allelism, such genes remain in the same population and are expressing themselves, however subtly or pronounced, in the phenotype, without changing the organism into a different species. Because when we talk about mutation, a immediate thought comes into our mind that if there is mutation, there would be genetic differences which will lead to variation. Variation might lead to isolation, and therefore the species will be separated into a different species. That is true when the mutations are so pronounced that they separate the individuals from each other and do not allow to breed. That is called evolution. But in multiple allelism, there is mutation, there is variation, but the genes remain in the same population. Because of variations brought out by multiple allelism, there is no breeding isolation. That is very important to understand. Only it gives us variety within the same population. So multiple alleles makes organism more diverse. However, it may be a prelude to evolution. It can in future bring such changes, but that is a different story altogether. It's not in the capacity here. Now, to end this, look at these 2 birds. The question remains, are they of the same species or are they different? This is the food for thought for you. Think and put in the feedbacks. Thank you.